Yeah, turn on. We can get started.
Okay. Uh, I welcome you all for today's talk. Today we have Professor Jinjun Shi. He is Associate Professor at Harvard Medical School. He did his PhD from Texas A&M University and his bachelor's from Tsinghua University. His research interests are fluorescence microscopy, nanomedicine, molecular biology, nanoparticles, disease biology, cancer, and he's a recipient of several awards uh, and has a great publication record in top journals like Science, Nature, and ACS. So I'll ask Professor Shi to really get started. Shi, over to you. Please, you may please start the talk.
All right. Well, thank you so much, Professor Mando. I'm so glad to see you again here, and was truly, it was my first trip to India a few weeks ago, and I was so glad to meet you and your over there. So I very much appreciate your invitation, and then I'd love to share some of our research work with you all here today, and so just a little bit of background introduction. So my name is Jinjun Shi. I'm from the Center for Nanomedicine at Brigham Women's Hospital, Harvard Medical School. Uh, hold on, let me have the laser point. Okay, so here is our research building at Brigham Women's Hospital, and then my office and my lab is actually on the seventh floor of this building. And if you will visit Boston, let me know, and I'll be happy to show you around. So, per Dr. Mondo's request, I am gonna talk some about cancer nanomedicine and some background of the cancer nanomedicine, and then some of our research work in this field. Um, so let me get started. So we all know that cancer nanomedicine is the application of nanotechnology for more effective, safer treatment of cancer. And this is a very exciting field, and certainly, it can be more broad, like include cancer diagnosis, cancer imaging by using nanotechnology. But today, my talk is more focusing on cancer nanomedicine for treatment. Right. So let me get it started. From 16. I'm sorry, and a lot of things happened in 2016, about 8 years ago. First one is the Binder Therapeutics, which is a biotech company, co-founded by my previous postdoc mentors, Dr. Amina Faloxat from Brigham Women's Hospital and Dr. Lob Delanger from MIT and other colleagues. And the technology is focused on the use of polymer nanoparticles, particularly like POGA and POA nanoparticles, and also coated with a targeting ligand to target the prostate-specific membrane antigen PSMA. And so Binder Therapeutics has been developing this technology and assume a lot of interesting data preclinically and also clinically, and they published several highly impactful paper in this field. And very unfortunately, in the middle of 2016, Binder Therapeutics filed a motion for sale of their assets. So the company literally went to bankruptcy, not because of the technology, but because of the many other issues. And then later, the company was sold to Pfizer for continuing development of this binders technology. And at that time, also after a few days after that, there was another news from Japan, also about pharmaceutical companies. They're developing new cancer nanomedicine called NK105, which is a polymeric micelles to deliver small molecule drugs, chemotherapy drugs for cancer treatment. In that case, it's a breast cancer in phase 3 trial. Didn't work well. And then right after a month after that, we had another news. It's about the Celulian, also a company in the Cambridge side in Massachusetts. And Celulian is a company, is developing also polymeric nanoparticles to develop small molecule drugs. And in the phase 2 trials of the CRLX-101, um, using the cyclodextrin-based nanoparticles to deliver a camptothecin drug. And in that trial, unfortunately, also didn't show better progressive free survival data compared with the standard of care for renal cell carcinoma. So there's quite some unfortunate, disappointing news from in that year. Certainly, not everything stuck in that year. Also, actually, months before that, this also liposomal nanoparticle for co-delivery of two drugs. One is the Nanomedicine, and then another drug is called cetabarin. So cetabarin and these two drugs, Nanomedicine, are co-loaded into the liposome, and then these vicositils, which actually showed promising data for instead of solid tumor, they actually used it for hematological cancer, AML, acute myeloid leukemia, and that drug was later on approved by FDA. But overall, 2016 wasn't a good year for this field, and very you may call this coincidence or what? I mean, that year you probably saw this paper, published by Dr. Warren Chan from University of Toronto. In Nature Reviews Materials, the title is Analysis of Nanoparticle Delivery to Tumors. And in that particle, they concluded this famous number called 0.7% of nanoparticle end up in the tumor tissue. So it's actually not as high as what we expected to the tumor after they reviewed our literature. Certainly that number is still debatable, and but that paper got a lot of attention. And a lot of people think the field for nanoparticle delivery for cancer treatment is was considered a hype. So right after that, we also was invited by the Nature Review Cancers to also write a review article on the same topic, cancer nanomedicine. And together with my previous supervisor, Dr. Amina Faloxat, and we certainly more focused on to understanding the success of this field, and then also why we didn't achieve what we expected in this very exciting field, and then the challenges and opportunities in the field. So both articles, received tons of attention, and then so far has been cited, over 3,000 times already. So today I'm gonna give you a bit of overview about this field, and then talk about a bit of the challenges what we recognized, and then the opportunities what we think we have for this field. So before 2016, we summarized these milestones in this field, 60 years ago, since the discovery of liposomal structure, and then the application of liposomes for drug delivery, and then right after that, also a lot of work from my previous mentor, Dr. Langer, for example, controlled release of polymer systems for small molecules and for micromolecules, and then the publication of long circulating PLGA packed nanoparticles 30 years after that, and then and then the approval, or first approval of liposomal drug Doxil by FDA in 1995, and right after that, 10 years later, we have another blockbuster drug, nanomedicine drug called Abraxane, which is albumin paclitaxel complexes, also received approval from FDA, and then between 20 years from first approval of doxil. We certainly have some other liposomal drugs that are approved, and then some like polymeric drugs received also approval in other countries, but not by FDA yet. And then inorganic materials like ionic nanoparticles also got approval in Europe for cancer treatment. And in 2015, the liposomal irinotecan was approved by FDA actually, and then now actually just right this year they received also approval by FDA for the first line treatment of a metastatic pancreatic cancer. So this is all the success we can see, in the field. But right, you know, you know, around that, like 10 years ago, we have been, slowly and then but steadily realizing the cancer nanomedicine field is not as simple as what we think. Give a nanoparticle can diffuse the cancer or some of the tumor tissue side, and then you have high accumulation, and you can cure the cancer. It's not that simple. So a lot of things we have a deep thinking, in-depth analysis of the field, and then that actually contributed a lot to our understanding of this area. And so that year, right after that, 2016, 17, we published this Nature Review Cancer paper. We have a very systematic analysis of nanoparticle systemic delivery to the tumor tissues. And the first thing I like to talk about is the EPR effect. This is a foundation, we consider as a foundation of cancer nanomedicine, the enhanced permeability of retention effect. You can take a look at this here, this schematic. And what we all consider as beginning with a nanoparticle after IV injection can circulate in the bloodstream and then get into the tumor tissues, like solid tumor tissues, through the leaky vasculature. And then because of the damaged lymphatic drainage systems in the tumor tissue, the nanoparticle can be trapped in the tumor site, and then release the drug and then kill the cancer cells. So we all know now this explanation of the EPI effect is very simplified, and then we have much more deeper understanding of the field. For example, how the nanoparticle interact with endothelial cells. It's actually interesting. Like Dr. Warren Chan's lab has published a paper a couple of years ago. They show that the nanoparticle can actually interact with endothelial cells, and then cross the endothelial barrier through the transcytosis in a much larger scale compared with the leaky vasculature. And then also my previous mentor's laboratory also, and then his colleagues discovered that the nanoparticle can be trapped by macrophages, and then increase the retention. So this is all like new understandings how nanoparticle interact with the microenvironment, with the stroma cells, and then even nanoparticle how they diffuse out of the lymphatic system is we are now having through the tumor tissues. So now we have better understanding, instead of just damage the lymphatic system. Indeed, the nanoparticle can actually penetrate the tissue and then diffuse out of the tumor tissue through the lymphatic system, still in the tumor tissue. So this is really we know the EPI effect. We have more and more understanding nowadays, and then that actually motivated us to develop the biomarkers for the EPI effect, because we all know now, and then there are publications in the literature, the EPI effect is not homogeneous. It's not as what we expect, leaky vasculature everywhere. It's actually very heterogeneous. And certainly, we can see from the clinical data, from the patient data, we know nanoparticle can get into the tumor tissues, but it's just not, very homogeneous. And then it really depends, the tumor stage and what type of tumors, patients, and it all matters. So it's very important that now we realize it's actually we need to develop a biomarker for the EPI effect. So I'm gonna talk about the about the efforts the field has done in a couple of slides, and it could be using imaging systems like imaging nanoparticles, or using protein biomarkers, liquid biomarkers, or to tissue markers, staining to really understand the biomarker for the EPI effect. And besides the EPI effect, and then we also have more understanding of nanobio interactions, because at the beginning, when you give the nanoparticle by intravenous injection, the first thing nanoparticle encounter is serum protein, right? So how the serum protein, they just don't, they tend to actually attached to the nanoparticle surface, really depending on the nanoparticle properties, right? The physical chemical properties, targeting ligand, and many other things. And the serum protein coated on the nanoparticle surface can actually dramatically change the in vivo behaviors of nanoparticles, including such as pharmacokinetics, secretion life, biodistribution, and tumor penetration is actually another challenge we have realized, in the field. We all know that nanoparticles tend to just accumulate around the tumor vasculature, like, captured by the tumor macrophage or trapped by the cellular matrix. So the nanoparticles are not easy to diffuse deeply to the tumor tissue. So how can we improve the tumor tissue penetration? There are also many papers published. You design using different nanoparticle strategies to increase the penetration. And after penetration, cellular uptake and the somal escape, particularly for RNA drugs, are also important. So these are all the things we have to keep in mind. And then one thing we don't really think about too much about is the drug release. Because we all know, if drugs continue to release or have a burst of release, then that won't work well to, for the therapy. And either you have to control the release or have a stimuli response of release that may work better. So these are all the designs. It's complicated, right? So here I just give you one example of the progress in the field for the EPI effect, and then was actually first published by presented in AACR media, and this is actually clinical data by Millennium Biotech, if I remember correctly, the company's name. And then what they did is they actually using iron oxide and nanoparticles for MRI imaging, and they give the ion oxide nanoparticle first, and then to look at the tumor tissue accumulation of this imaging agent, and then they then give the therapeutic drugs like liposomal drugs, and then look at the response of these tumors that have different level of ion oxide nanoparticle accumulation, like flumoxetine accumulation into the tissue. So you can find if you have higher accumulation of the ion oxide nanoparticles, the response of the tumor tissue to the liposomal drug is actually much better compared with these tumor tissues that has lower ion oxide nanoparticle accumulation. So that actually indicate that the possibility of using imaging agents like ion oxide nanoparticles to screen the patient or to select the patient. And right after that, my previous supervisor, Dr. Amit Falakzad, and then his collaborators, Dr. Ralph Weislet from MGH, they actually tested this, this idea, the concept, in animal models. They use the ion oxide nanoparticles as an imaging agent, and they use the PLGA packed nanoparticles, and they injected these the ion oxide nanoparticle first, and then look at the different accumulation, and then to look at the also the response of the tumor tissue to the PLGA packed nanoparticle loaded with docetaxel. So you can see here similarly, at high accumulation of imaging agent, you show the best response of these PLGA packed nanoparticles. But with the lowest accumulation, you see rapid progress of the tumor size. So all this shows like an imaging agent could be or other biomarkers actually could be important to maximize the therapeutic value of the cancer nanomedicine. So certainly there are many other progresses in this field. There are many good publications, review articles. You can take a look. So so then I'm gonna switch my topic from small molecule. What I talk about is mostly small molecules, and then what I talk about is bigger molecules like RNAs. So that's why my title is called from small molecules to RNAs, and again, right around like 2016, I think it's 2018, we have the first siRNA drug approved in the field called Onpattro, using liposome to deliver siRNA to the liver, and this is for the genetic disease called amyloidosis. And and then a couple years ago, 3 years ago, now we have the messenger RNA vaccine, also using lipid nanoparticle for the vaccination and for COVID-19. And early this year we have first FDA approval for using genome editing for sickle cell disease treatment, which is they did ex vivo stem cell engineering with the genome editing and then refuse the stem cell back to the patient. So RNA medicine are now becoming real. So we all know, and then this has a huge actually impact for cancer treatment also, and but before I talk about it, let me just give you a brief review about what these messenger RNA is, which I'm going to talk about quite a bit in the rest of my talk. And mRNA was actually first discovered around 1961, a few years before the liposomal structure discovery. And very interestingly, nobody received the Nobel Prize for the discovery of mRNA structure. So there was a lot of quite interesting scientific story behind that, indeed. And the mRNA, as we all know, is actually transcribed from our genetic DNA, and this carries the gene information, and then it can be translated into the protein. So compared with DNA, mRNA, as a therapy, could have some potential benefits. For example, you don't need to deliver the mRNA to the nucleus, so you only need to deliver to the cytosol, right, for the translation. So there will be no nuclear barrier, and then you could also avoid the potential host genome integration. And mRNA also have a rapid and more regulable protein expression compared with DNA. So that's why the mRNA, when we overcome the synthetic manufacturing issues, then it rapidly becomes a very hot topic as a therapy, and certainly that was exemplified by the approval of the the COVID mRNA vaccine a few years ago for pandemic. And this vaccine, two vaccines, one's by Moderna and right in the Cambridge, the other one's by Pfizer BioNTech. This vaccine has mainly two components. One is the mRNA, which codes the spike protein, and the other one is the nanoparticle. And in this case, it's a lipid nanoparticle. So why nanoparticles are important in this case? That's because mRNA is big and it's highly negative charged and it's susceptible for degradation. And you also need to get into the cytosol for protein translation. So nanoparticle could solve all the problems, and then that's so what we consider it's equally important, for the success of the mRNA vaccine. And then right after success, certainly that motivated us to if you look at it a few years ago, 5 years ago, I don't know how many labs are working in the field, but probably not a lot. But now you can see many labs are now working with mRNA delivery or mRNA therapy. So well, actually cancer using mRNA for cancer treatment is actually much earlier compared with the success of compared with the development of mRNA vaccine for COVID-19. Like the companies CureVac and BioNTech and Moderna, they all started clinical trial way earlier, compared with the COVID-19. And so far in the field, although we don't have any FDA approval yet, but there are many in the clinical trials, including many from the Moderna, BioNTech, and CureVac. They are developing cancer vaccines using mRNA. It could be some specific tumor antigens or personalized neoantigens. So and then recent clinical data from Moderna shows like cancer vaccine, neoantigen vaccine actually works pretty good in combination with immune checkpoint blockade therapy. Meanwhile, also there are clinical trials, for cancer immuno-oncology. You can, use mRNA to code like cytokines or other, inflammatory factors that can actually improve the immune response in the tumor microenvironment, and then improve the immunotherapy. So these are a few clinical trials, and then this is a review article written by my colleague Dr. Izzo Dom a couple of years ago. And so my lab actually started working with mRNA. Actually, it's over 10 years ago already, and I still remember that was at end of 2012, and I saw a report published by Boston Global talking about a company called Moderna, right out of the stealth mode at that time. And I read an article. Certainly, my professor Dr. Langer was a co-founder for Moderna, and his name he mentioned about this company, and I saw that the report. I was very impressed because they want to use messenger RNA to generate a new protein, or to restore lost protein. So at that time, my lab was working with siRNA, small interfering RNA, for gene silencing for cancer treatment. When I saw the picture, I was like, dude, that was great, because on one hand I can use siRNA to silence the gene, but on the other hand, we can use messenger RNA to restore some lost genes, right, like tumor suppressor. So if I combine these two technologies together, then you probably, which are very complementary to each other, you can literally almost do anything you want to modulate any gene, right? So that's how my lab actually started working with the messenger RNA, 10 years ago. So as you know, cancer is highly involved with genetic mutations, and particularly the overexpression of like a lot of oncogenes, pro-oncogenes, and simultaneously loss of mutation or loss of function of tumor suppressor genes. And then these the loss of a balance actually can transform the normal cells into the tumor cells. And also these genetic mutations promote the invasion of tumor cells and metastasis, and also contribute to the drug resistance. So as in the pharmaceutical biotech field, and most drugs are developed to target this overexpressed oncogene, right? And unfortunately, there are few options that can be used to restore the lost tumor suppressor. So we really think that's the opportunity for messenger RNA to restore tumor suppressor. This is how my lab actually started working with messenger RNA and then to restore tumor. And before that, actually, we were actually also working with the messenger RNA for cancer vaccine too. But today I'm more talk about using messenger RNA to restore the tumor suppressor. So here are two actually well-known tumor suppressors. The first one is the P10, called the phosphate and tension homolog deleted the chromosome 10. And this tumor suppressor is actually quite widely mutated in different types of cancers. For example, in prostate cancer, as you can see here, about 40 to 50% of later stage prostate cancer patients, they have a complete loss of a P10. And the loss of P10 not only contributes to tumor growth, but also the drug resistance, like to small molecules, as published in the Nature paper, or to the immune checkpoint blockade therapy published in Cancer Discovery, which I'm going to talk about later on. And the other one we have studied is the P53, which is the most widely mutated tumor suppressor gene in many cancer types, right? So you can see here. And then this P53, as we all know, is a master gene regulate and controls many biological pathways in the cells, as well summarized by this review article in Cell by Dr. Scott Rowe and then his colleague. So To deliver mRNA to solid tumors is much harder compared to the just give the muscular injection of mRNA vaccine. The reason is because of the IV injection, as I mentioned earlier on, right? For the cancer nanomedicine, all the the delivery uh, issues, and because there are multiple challenges involved. For example, we need to protect the, you compare with mRNA, you even protect more of mRNA because you don't want mRNA to be degraded before they reach the tumor tissues. So you have to encapsulate the mRNA, in the nanoparticles, for example, and then you need to have a relatively long circulation for the mRNA to, or siRNA or other RNAs to reach the tumor tissues. And then as the EPR effect, you want to, how can you increase the tumor accumulation? That's another challenge. And penetration is certainly, a very important one. And compared with the small molecules, you probably don't really need the nanoparticle uptake by tumor cells, because small molecules can just release it from the nanoparticles and then diffuse into the tumor cells. But for RNAs, like particularly for like messenger RNAs, big RNA, they can't get into the tumor cells. So you have the nanoparticle have to carry the mRNA or other RNAs into the cells, like through either with ligand receptor interactions or just non-specific cellular uptake. And after internalization, endosomal escape is critical for RNA therapy, because if you just end up in the endosome or lysosome, don't get it into the cytosol, then siRNA, mRNA, or other RNAs won't have the bioactivity. So over the years, my laboratory has developed multiple different nanoparticle platforms, from the hollow lipid polymer hybrid nanoparticles to these solid nanoparticles to make it much smaller. This is a hollow structure, has like probably 150 to 200 nanometer. And with the smaller ones, we can reduce the size to 70 to 100 nanometer. And then we also added some new functions to the nanoparticles, either targeting ligand or stimuli responsive component. And also we are now developing a new generation nanoparticle, including lipid nanoparticles. The whole purpose for us to do this is to solve all these multiple static challenges in the systemic RNA delivery to tumor tissues. Like how can we improve circulation, better tumor accumulation and penetration, and how can we increase the cellular uptake, not only the tumor cells, but it could be also other stromal cells like immune cells in the tumor environment. And the somal escape is very important. Also, we are also interested in the durable activity by controlled release or other mechanisms, because we all know RNAs, like particularly for mRNA, they are transient. So if we can prolong the activity of RNAs, then you can give a lower frequency. The dosing could be lower frequent. So I'm going to more talk about just these solid lipid polymer hybrid nanoparticle platform today. And this nanoparticle is actually very similar to the lipid nanoparticle. The difference is, we put, added some polymers into the core of these nanoparticles, so with the hope of to increase the stability of nanoparticles and other adding on new functions like, co-delivery of drugs, other drugs, and the controlled release. So this is a two-step, robust self-assembly. You first mix these mRNA together with a ionizable lipids like the G0C14. We synthesize in the lab and then also co-dissolve in the organic solvent, the DMF. And then we add these to these organic, water phase containing like lipid pad, and then polymer like POGA, which is hydrophobic, which is precipitated out to form nanoparticles. And meanwhile, this lipophilic lipid with mRNA complex can also co-precipitate out with nanoparticles, trapping the nanoparticles. And then meanwhile, these lipid pad can self-assemble on the nanoparticle surface to stabilize the particle. So these particles shows, as we did confirmed, enhance the cellular uptake and endosomal escape, and also prolong that PK, and then improve the tumor accumulation. And it shows the size around 100 nanometer, and they are stable. And with these nanoparticles, we load the P10 mRNA and then incubated with the P10 mRNA nanoparticle with the P10 lost human prostate cancer cell line like PC3, and we can see the very beautiful translation of the protein after day or two, and then that actually induced the apoptosis of the cells through the PI3K-AKT pathway inhibition. And this particle also showed the dose-dependent toxicity in PC3 cells, and also another P50, P10 lost tumor cell line, the LINCAP cell line, prostate cancer cell line. Meanwhile, we also tested this nanoparticle in prostate, normal prostate epithelial cells, and also P10 competent prostate cancer cell lines like DO145. And in both cases, you can see we show the much better cell viability compared with the LINCAP or PC3 cells. So we suspect, then this strategy could be specifically used like for precision nanomedicine to only target like the P10 lost tumor cells. So we then did the testing of this nanoparticle in vivo of the intravenous injections. Every 3 days, we can see the subcutaneous tumor growth inhibition in mouse models and the P10 expression in the tumor tissues and the better survival. We also tested this in a bone model of, prostate cancer. As we all know, around 80% of the later stage prostate cancer, they have bone metastasis. So it's really critical if we can target the tumor cells in the bone tissue. And then that's what we did, after, injecting these, we implanted these tumor cells through the tibia of the mouse, and then this PC3 cells express the luciferase, so we can use the whole body imaging systems to look at the monitor tumor growth by imaging the luminescence signal. And after 4 cycles in IV injection of our nanoparticles, we also similarly see the inhibition of tumor growth in the bone tissue. So this is what we have done in the human prostate cancer models. And when we were working on in that paper, and we read a few papers published by clinicians. These are the papers with published data with clinical studies from clinical studies. For example, like the first one is the cancer discovered by Dr. Patrick Hu, and then what they found is they look at the P10 levels and then in the tumor tissues and how that correlated to the immune therapy like immune checkpoint blockade, like PD-1 inhibition. So what they say is very clearly, in the presence of the P10, you can see better. You can see tumor response, shrink, tumor size reduction after the immune therapy treatment. But in the absence of P10, you can see the tumor growth. And meanwhile, they also see the P10 presence actually can. You can see more, CD8 cytotoxic T cell infiltration to the tumor tissue. And after that, there's a few papers published in different models. This is a melanoma model, by the way. And then we also published, discovery in other types of cancer like breast cancer and lung cancer. All this clinical study shows like a very consensus conclusion is P10 loss is actually associated with the immune suppressive tumor microenvironment, which decreases T cell infiltration and then the overexpression of immune suppressive cytokines. And all this contributed to poor response of the tumors to the P10 lost tumor to the immune checkpoint blockade like anti-PD-1. So we were very excited when we saw this paper, and then we have this nanoparticle, P10 nanoparticle. So what we all need to do is just switch the human prostate cancer cell line to mouse cell line, right? Like for example, P10 kappa A, which is a prostate cancer mouse cell line with P10 loss, and B16F10 is the melanoma cell line, mouse cell line with the P10 mutation. So in both cases, you can see the dose-dependent toxicity, but more interestingly, we saw that with the cell death, the tumor cell death, we also see very dramatic change of the immune genetic cell death biomarkers like characterizing protein expression in the membrane, the release of ATP and HMGB1, all this indicating, the cell death induced by P10 restoration can actually um, trigger the immunogenicity. So we did also other biology, although we are not a biology lab, but try to understand how this P10 restoration trigger the immune response. So we think that could be through the pathway, called autophagy pathway, because P10 actually can inhibit the PI3K AKT mTOR pathway, and the mTOR pathway can activate the autophagy pathway. So for example, if you add these 3-MA, which is autophagy inhibitor, to the P10-MI nanoparticles, you can see the reduction of the ATP release and then the reducing of the calreticulin protein expression on the membrane, cell membrane. So on the other hand, if you combine with the autophagy activated like a rapamycin, the combination of P10-MI nanoparticle with rapamycin actually can induce more cell death. So then we moved on to test whether P10 restoration can actually moderate the microenvironment in vivo. And that's what we did. We did a 3 IV injection, and then we sacrifice the mice, take the tumor tissues, test the immune cells and also cytokines. And then here you can see the affected CD8 T cells have higher level expression in the P10-MI nanoparticles. And meanwhile, we can see the reduction of the immune suppressive T cells, like regulatory T cells, and then also the myeloid derivative suppressor cells in the tumor tissue. So we also did a lot of cytokine testing to show all of this data support that P10 restoration can actually improve the immune activation. And then we combine the P10-MI nanoparticles with PD-1 in this B16F10 subcutaneous model, which doesn't respond well to the PD-1 therapy. And actually, it will give you just a P10-MI nanoparticle. You do see the slopes of the growth, tumor growth, but the combination definitely shows the best of the anti-tumor effect. And then we also confirm that the CD8 T cell response is actually from the P10 itself, not from anti-PD-1. And we also depleted all the T cells, and then we give the challenge the mouse with the tumors, and then you can see with the tumor with the T cell depletion, the tumor grows even after the P10-MI and anti-PD-1 combination treatment. The growth of the tumor is is actually restored in a certain way, similar to these just P10-MI nanoparticle in the subcutaneous model. So this indicating the importance of the T cells for this anti-tumor effect. And so that's what we did with the P10. So we were thinking, whether we can expand that to other tumor suppressors like P53. And we collaborated with Dr. Dan Duda from MGH, and using his orthotopic model of hepatocellular carcinoma (HCC). And then, by the way, the immune therapy was over the last couple of years. It was actually just approved as either first line or second line treatment for HCC. But unfortunately, around only like 20% patient HCC patient have a good response to this immunotherapy treatment, and majority of them they still don't respond well. And meanwhile, we all know a third of patients of HCC they have actually P53 mutation. So we're thinking whether very interesting to understand whether the combination of P53 with the immune checkpoint blockade, whether that can actually have a better anti-tumor effect in this model. So similarly, we use these the hybrid nanoparticle platform. Certainly, we did a lot of optimizations. For example, like GCLC14, we found that GCLC8 actually works better than GCLC14. We're also adding targeting ligand, to the HCC, a peptide called the CTCE, which you can target the CXCR4 receptor on the tumor cells. And when we added only 6% of the ligand density, it doesn't change for the PK, but it definitely improve the tumor accumulation as compared with the free mRNA or the non-targeted mRNA. So we did a combination, and you can see here that definitely shows the better response compared with just the PD1 or the P53 nanoparticle. And then we look at the tumor tissue again, look at the immune cell response, cytokine response. Actually, very different from what we saw from P10, which by itself can actually moderate the immune microenvironment, but P53 in these orthotopic models by itself does not. So only the combination of the P53 with the anti-PD-1, we can see the dramatic change of like T cell infiltration and CD4 T cell infiltration and like NK cell infiltration, and then also the expression of the cytokines like TNF alpha for this combination group. So we don't fully understand yet why this is the case, and we are doing a lot of biological studies together with Dr. Janduda, and then hopefully we can have more understanding of this field. So then we did a longer term anti-tumor efficacy studies, and then after 5 dosing or 4 dosing, I can't remember, 4 dosing probably every 3 days, and then we just let the tumor grow. So you can see here, this combination definitely shows synergistic effect compared with the monotherapy of either by anti-PD-1 or P53, and that actually improved the tumor survival, also reduce the lung metastasis and other complications like ascites, in the abdomen site. So this actually shows the another story. Shows the using messenger RNA can restore tumor suppressor, and then for the treatment, and combine with immune checkpoint for cancer treatment. So hopefully, I give you a good overview of, this field, cancer nanomedicine field, and then as well as some of our recent work. And I hope I convince you the cancer nanomedicine field is very promising. It's exciting. Although we see a lot of challenges now, but, and then need a more fundamental understanding of a lot of things like the EPI effect, the development of biomarkers, nanobio interactions, right, and drug release. But we think, with more and more understanding of these the fundamental issues, and then we think, cancer nanomedicine could become a mainstream, future main therapy for cancer. And certainly we need to discover more better biomarkers for the EPI effect and for patient stratification. And also, meanwhile, RNA nanomedicine is emerging for cancer, many clinical trials as a cancer vaccine using lipid mRNA nanoparticles, or immune oncology, for the tumor injection, and then other things like using messenger RNA or to engineer the T cells or NK cells to in situ engineering or ex vivo engineering to have a T cell therapy or other immune cell therapy. And then in our lab, we showed that mRNA can be used to restore tumor suppressor like P10, P53, and then in either case, we can see the combination of P10 or P53 with anti-PD-1 can give you much better anti-tumor effect. And we have also did some other studies like restore P53 can actually improve the sensitivity of some chemotherapy drugs like mTOR inhibitor to P53 lost tumor cells. And then there's actually also interesting work we are now working on is we found out the combination of the tumor suppressor with some certain immune stimulators. We can actually very selectively target some certain immune suppressive cells in the tumor tissue. So with that, I think that's about what I'd like to share with you today. And then I'd like to acknowledge my collaborators like Dr. Bruce Zeta, Dan Duda, Chuck Beveridge. We work together for the cancer immunotherapy. And I didn't get a chance to talk about some other works in my lab, like working with Dr. Eric Tabase from Tabas from Columbia University, Dr. Hong Chen from Children's Hospital. We were actually using RNA nanoparticles to target atherosclerosis. And with Dr. Stacy Kota from also Children's Hospital, we are working with the genetic disease. And Arif contributed a lot to the first P10 mRNA delivery work. Now he's a principal scientist and also a project lead in Sanofi mRNA Center, doing mRNA vaccine now. And Dr. Yaoxing Liang did the combination of P10 with immunotherapy. He's now faculty. And Dr. Yulin Shao is instructor in my lab, and then she did all the P50 work. And Dr. Yudong Som is actually now she's working on the combination with the tumor suppressor with immune stimulate. And I also like to acknowledge the funding support, and which is very important to the lab. And my last slide I would like to show you is follow again from Moderna's picture. I'd like to show you this RNA therapy is just the beginning. The COVID-19 success is just the beginning. And that's just because mRNA, you can do a lot of things. You can use that to generate intracellular protein, transmembrane protein, secreted protein. You can use that to restore the endogenous protein. You can use that to generate the new protein therapy, like engineer the proteins, antibodies. So there's you can even use mRNA as a adjuvant or many other things. You can use that to engineer cells. So a lot of opportunities in the field, and I think it is going to have a very, very exciting future for cancer and other diseases. So with that, I'd like to thank you for your attention. Stop sharing my slides. Thank you. Happy to answer questions.
Yeah, indeed. Thanks a lot. Thank you so much for delivering such a wonderful talk and a nice walkthrough through the development of nanomedicine and RNA medicines. I guess we are entering a new era in medicine, as far as I can understand from your talk. And I guess this is the first such talk that has ever, I think, it has happened in Indian subcontinent. I mean, I have not heard of nanomedicine or mRNA kind of medicines so much. And I really thank Professor Jinjun for introducing us to this emerging field of nanomedicines, which we are going to see in future more and more. The talk will remain in YouTube for researchers around the world, and this is going to benefit a lot of labs and researchers, emerging, researchers as well. I think we'll just have a look if we have some questions online, because this is YouTube live. So we want to see if there are some questions online. So Arvind, do we have some questions?
No, sir.
Okay. Okay, fine. Anyone of you want to ask any question? Then we can go ahead with that.
Yeah, feel free to ask me questions. I think I did, I hope I did
have one question.
I explained it well for what I want to say, and then but feel free to let me know if you have any questions.
Yes, I have a question. Can I?
Of course.
Yes. So, does the size of these nanoparticle matters in drug delivery? Do we need to take care of the size? Does it play any role?
Yeah, I think that's a very good question indeed. So size definitely matters, and there are actually not just my lab, but many other labs have published. Like for example, Dr. Kazunori Katoga's laboratory, they published a paper for it really depends on the also cancer types. So for example, for the pancreatic cancer, which we know have less vasculature but more dense, actual cellular matrix. And yeah, the size, within the paper published, is smaller is better for the penetration, for the accumulation. So also, size matters. For vaccine also, even subcutaneous injection, smaller size can actually penetrate better to the lymphatic system, to the lymph nodes, compared with the big nanoparticles. So all these, I think, it matters for cellular uptake, for, as I mentioned, for penetration. So I do think size is important. But meanwhile, if you make the particle too small, right? I mean, then you have to, keep that in mind. Smaller particle, then the release could be faster, right? Like for example, for small molecule drugs, right? So how do you balance these? All these issues, I think it's important, for consideration. So, but I think it depends on case by case to study that. I mean, smaller particles certainly, I think we're working on that also. Try to make a very small RNA nanoparticles, and then hopefully then that will give you a lot of advantages. If you want to target some unique tissues.
And one more question, Professor. will it have any disadvantage or side effects, something like that? This nanoparticle based drug delivery.
Well, I mean, of course, I think I would like to see probably all medicines has toxicity, but it's really about the therapeutic window, right? That's why we have to do a lot of clinical trials like phase 1, phase 2, and even phase 3. So phase 1 was more focusing on the toxicity, and so that's how you identify the therapeutic window to pick up the dosage that is has tolerable toxicity to patients. So for I like to point it out is um, for RNA therapy, what we think could have some potentially the combination with the immune therapy. We think that will give you more opportunities to have even potentially safer drugs, because if you want to design this mRNA response, or the nanoparticle can be delivered to only like selective tissues that could potentially reduce the toxicity. And certainly that also is true for not just mRNA but also for other drugs, right?
Thank you, sir.
Jinjun, I have a question. And what are the key factors that determine the permeability? Does pH has a role to play? And are these nanoformulations better targeted than traditional medicines?
Well, yeah, it's a great question. So if I look at that, uh, a lot of some traditional formulations are way, like for example, chromophore, right? For the paclitaxel. It's just we never called it nanomedicine, but indeed they actually form nanoparticles. It's just we didn't realize it. Some traditional formulations, right? But with more and more understanding, we know, uh, nanoformulation certainly could have a lot of benefits, but meanwhile, it could also induce new toxicity, like dog cells, right? It reduce the cardiotoxicity, but induce the foot hand syndrome, right? So that kind of limits the application or limited dosage of the also as I mentioned earlier on, we had a lot of high hope of this candidate. We feel like, oh, that could solve a lot of problems, like reduce the toxicity, increase the tumor accumulation, cure more cancer cells. But at the end of the day, from all the clinical trials, what we saw is it's not that simple. So we do need a more fundamental understanding, particularly in patients, I think.
Okay. I mean, if there are any questions, you people can ask. Yeah, we have a question here. Yeah.
Okay.
So it was quite an interesting talk regarding mRNA. But when we study regarding mRNA, we find that the extraction of mRNA and that the process mRNA to protein translation is quite short period. So how do you perform all such manipulations before the translation process? Isn't it a very challenging task?
I think it is. You're right. Absolutely. First of all, that's the nature of mRNA, right? It's short. Half-life of mRNA itself is not long, right? So you get probably a short translation, like in general, you see protein expression reach a peak, 12 hours to 24 hours, and then the protein starting, expression starting to decrease, and within a few days, your protein is gone, right? So there are ways, people are trying to improve the mRNA stability, like through chemical modification, or through the poly A, for example, modification. Then the enzyme won't be able to cleave from there, so that can increase the endogenous stability, and to prevent the enzyme degradation, endogenous enzyme degradation. And there are other ways, like for example, you can make the linear RNA to become circular. They call it circular RNA. And then the circular RNA can certainly have more stability compared with linear RNA. So those are all the possible ways to improve the stability of RNA and then improve the translation and then duration. And nanotechnology, on the other hand, is also a possible way to do that. And my lab is working on that also. And how can we use nanoparticles to improve the stability of mRNA and control the release of mRNA, right? And this all could, lipid nanoparticle, by the way, doesn't do much of that. So, I mean, traditional lipid nanoparticles. So that's actually, I think is the opportunities for nanotechnology, right? To solve the problem you just mentioned about. So I think there are a lot of things that we could do. It's very exciting.
Yes. Thank you so much.
Thank you.
Anyone want to ask any more questions that you may have? I don't see. I mean, thanks a lot.
That's perfect.
Yeah,
Keep time 1
Let's
hour.
thank, let's
And
thank the speaker.
thank you very much. Thank you
Yeah.
all. And Dr. Mondel, appreciate your invitation. And then really, I'm looking forward to see you next time. And if you visit Boston, let me know.
Right.
And, okay. Wish you have a nice Saturday over there. And we'll see you next time.
Okay. Thanks a lot. Thanks a lot.
Bye.
Thanks a lot.